Hello.
Hi, Aneesh. How are you?
I'm good, and you?
Sorry, I was. Yeah, good, good. Thank you. I'm trying to put up the topics. Do you see on top science biology? And now I'm
Yes.
searching for ecology or environment. There's
Yeah.
environmentalism.
Environmentalism. Yeah.
Yeah, maybe.
Is
I'm
that,
here. I'm here
yeah,
with
there's not
Aaron.
too many options. There is environmentalism and then, oh, yeah, that's it, I think. What? There's no ecology. Okay, that's probably the closest we can get. I'm sorry. There's like for science, it's
Yeah.
very
No, that's
limited.
no problem. Environmentalism and biology covers it pretty well.
Okay. How are you?
Um,
Sorry.
I'm here with Aaron as well. Both of us are on the same microphone,
Yeah.
Ah,
I guess.
okay, great. Perfect. Good. Yeah, that works. So welcome both. Welcome Aaron too.
Thanks.
I'm sharing now the document so we can check if
Okay.
it's working. Here, let me add it.
Okay.
No, I'm just.
There. Do you see it? Should be there now.
I see it. Yeah.
Perfect. And now I will share your websites in the chat for everyone so that
Okay.
they
Perfect.
can. Okay. I'm sorry, there are things I can only do once the room has started, so I squeeze in a lot of. And then, Erin, you have. I found two. I found the one that's your own, or the one that is
The
by.
work one.
Yes.
You can use the one that's my own. My work one
Okay.
just directs to the one that's my own.
Okay, perfect. Yeah, so I did that. And I will
Sure.
add the paper. The paper link. Sorry. For that's why I start earlier so we get all of this done before the actual start time. And welcome everyone. Nice to see everyone here. And yeah, we'll start in around 5 minutes. So we have a few minutes, which gives me time to say on Twitter that we are about to start too.
Cool.
And yeah, thanks for doing this, for coming here, making the account and everything. Did you, since you made the account, try out Clubhouse a little bit or,
I haven't. To be honest, I'm a little naive when it comes to the uh, social media technology. I did try listen to a few other podcasts that had been posted from previous speakers. Um, so I did listen in on a few of those, um, but I'm still learning the app. Let's put it that way.
Oh yeah, that's totally fine. I was just checking in general if it's, I listen to, I used to, when the shutdown was, people used to be here, almost all day long.
Oh yeah.
And so you can, there are music rooms, there are yoga rooms, meditation rooms, political rooms, news rooms, or
Cool.
clubs, and
Hmm.
different science clubs and book clubs, and anything you can imagine you can find on here. So yeah, you can easily spend your whole day. And then when the Ukraine war started, there was an ongoing room for
Oh yeah.
months that had like millions of people in the end, I believe. participating over time, so it was 24 hours.
Mhm.
Then there's an Iranian room to keep us updated about what's happening there, and you know, all kinds of
Yeah.
stuff. You
Well,
can always be on here.
it's something that we're exploring on it. Um, it's pretty new to us, but it's very cool. Sounds
Yeah.
very cool.
Yeah, it used to be way more busy since people were just, stuck at home.
Hmm.
So yeah, but yeah, there's still a lot going on. Then, especially if you want to listen to people in different countries, what like their viewpoint is, how news are perceived there, and so on. I think that part is really interesting if you're interested.
Hmm.
And the one that I usually listen to and comment on is tech news, but they moved to Twitter Spaces. Um, Twitter has something similar, but I feel for recording sessions like ours, Twitter still has a lot of issues, bugs, and I'm not willing to deal with that for
Yeah, I
moving
agree.
to Twitter.
Especially this seems to be a very busy uh, busy chat room, or I guess you have multiple speakers on every week, and so there's always something happening, right? You need a platform that can accommodate it.
Yeah, exactly. That works like all the time. It would be I feel like a disaster if, we spend, I don't know, some time like a month or two to meet and plan and the app wouldn't work. Like,
Yeah.
then that would be to find another time again. It would just be very embarrassing. So I'm not
How
willing to.
long has this been going on, Science Society on Clubhouse?
So that I made the own club out of it, it's
Mhm.
pretty a little bit over a year.
Okay.
Before I just did it in other clubs or just, you know, randomly, not so planned. What drove me to create this very own club was that, like when I first came here, I was pretty shocked. Like I was on social media before a lot and stuff like that. I was pretty shocked at like, how negative the idea was of people of science and scientists, how we are and all of that. And I wanted to, and people asked me a lot of questions of fields that I don't know much about. So I started to invite people that actually, know what they're saying because they work in the field. And then in other clubs, this would then get mixed up with people talking that really don't know what they are talking about. And I felt like I would just contribute to that fake versus actual knowledge by mixing that up, kind of, if you know what I
Mhm.
mean. So to give legitimacy to people that don't know at all what they are talking about by having also people on that are actual scientists. That's why I then created, also was one of the reasons I created the own club. So when people come here, they know I invite the people that are actually working on this and not just people pretending to know about the field. So, anyways,
Right. Yeah. I mean, it's a great idea. We're certainly on board with it.
oh, thank you. Yeah, and it's 12, so we can just start.
Hmm.
Um, I think people will still be coming in, but let's start with introductions, and then we'll go from there. So, welcome everyone to Science Society, and a special welcome to you, Anish and Eric. And before we start, let me give the audience a short introduction so they get to know you a little bit. So,
Hmm.
Dr. Aneesh Bose, he is a postdoctoral researcher in the Department of Wildlife Fish Environmental Studies at the Swedish University of Agricultural Sciences. and he did his bachelor in science in marine and freshwater biology at the University of Guelph, if I say it right, in Canada.
Yeah, well,
And
that's
and PhD in psychology and neuroscience and behavior at the McMaster University in Canada, with in the lab of Dr. Siegel Holstein.
Sigal. Yeah.
Oh, okay. Thank
Hard
you for
names.
correcting.
Sorry, what's all right.
No, I'm glad you're correcting me. And then he did different postdoctoral research, but let's get into that with the interview later on. And Dr. Erin McCallum, she's a behavioral ecologist and ecotoxicologist. And she is really interested in understanding the causes and consequences of anthropogenic stressors for aquatic life. And she would like to, with her research, to provide like a scientific and informed policy for conserving fish, aquatic habitats, and freshwater resources. And she is an assistant professor at SLU in Umeå, Sweden. And yeah, as I said, you can check out the bios in on the website that I shared. And hi, Victoria. You made it. How are you? I'm sorry to interrupt, but Victoria is our co-moderator, and meet Aneesh and Aaron here that will be presenting today.
Oh, thank you so much. Good morning, everyone. Good morning, Katarina, Anish, Aaron. Does Aaron have their own separate PTR, or do I just not see it on my screen, or are they
Um,
sharing?
Yes.
Aaron and I are both sitting together at the same
Yeah.
microphone.
Okay.
Yeah, we thought it would be
Good.
easier than just sitting like down the hall from one another.
Yeah.
All right, fantastic.
Victoria, I know you just arrived. Do you want me to ask the interview questions, or do you want to go ahead? Both is
I
fine.
am prepared and I'm ready to go.
Wow,
Even though
perfect.
we have lots of snow outside. I just have to announce that I'm originally from California, so whenever it snows here, it's an enormous, exciting event. So anyway, yes, I am completely ready if everyone else is.
Yeah, yeah. We are ready to go at your
All right,
pace.
thank you. And please remind me, we have Anish, and who else do we have?
Aaron.
Erin. Okay, hello. Hello. Okay, so great. This question can be for both of you. So welcome again to Science Society. We're so grateful that you're here to share your work with us. And the question is to give us a personal side to the guest speakers and maybe learn a little bit more about what drives you toward your research and even drives your research. So when do you feel that your passion for science developed, and or when did you first notice that initial spark? And this could be in your childhood or anywhere in your life.
Um, should I start?
Yeah.
I can start. Yeah, that has been there since the beginning for me. I always wanted to work with animals. I think that was a real passion of mine since as long as I can remember. I always was drawn to uh, aquatic animals in particular. I'm not sure why, but in fact, when I look back at a lot of the old finger paintings that I made when I was in preschool or daycare, I was drawing pictures of fish, and so for some reason I was always really interested in aquatic animals, and I really wanted to work with animals um, as a career. So I actually forgot a little bit about that passion at the end of high school when I was trying to figure out where I would go for university, and I wound up starting out in engineering. I did a year of that, but found myself uh, wanting to do something a little bit more um, closely connected to animals and nature, and so I wound up switching to marine biology. And so it was really like, yeah, a rediscovery of my early childhood interests. And then I followed that path to where I am now.
Yeah, and for me, I was always an animal lover when I was younger, and loved being outdoors. But I didn't really get into science until I was in university. I originally went to university to be a psychologist, so I was doing a Bachelor of Arts originally. But then I took a class called the Biological Basis of Behavior, which was basically an introduction to neuroscience. And that's where I really got interested in the sciences and doing research to ask and answer questions. And so I ended up switching my degree from a Bachelor of Arts to a Bachelor of Sciences, and then followed the research focused career path to where I am now.
Thank you very much. It's especially wonderful to hear both of your answers together because
Mhm.
some so you have so the similarities and differences that you both switched and went to where you needed to be for this moment in your lives, and also that Anish, that you had that initial affinity toward
Yeah.
marine, the marine environment, and it's such a beautiful feeling when when you recognize that, and then you don't argue with it, and then because you can't, it's something innate, in us, I believe. At least that's what I believe for myself. But I hear these when we hear these stories and we ask our guests, and then they say, well, I had this feeling, and then I tried to be an electrician or something, and then I realized that
Yeah.
what I really needed was neuroscience, and so here you are. And so what I always wonder is, what is it that helps some people listen, and gives some people the power to act on that, and others, there's just a longer path, or maybe don't ever align with that feeling. So bravo to both of you. And then my next question before you dive into your presentation is, if you can take us maybe through some of your favorite jumps along the path that brought you from that moment, or that study of the research that you chose and eventually up until now.
Oof. It's been a lot of bouncing around. Um, so from my undergraduate time, I always was seeking out opportunities to get research experience in different labs. And so I was always asking professors if I could volunteer in their labs or help out on a field project. And so I wound up getting a lot of experience in a variety of different places. So I've worked in some uh, labs where I went out to the field to collect insects in lakes and ponds. I've worked in labs where I got to go on research vessels in the North Sea, or the Weddell and Scotia Seas. I've worked in labs where I could do field work in lots of different places of the world, like in different parts of Africa, or Mediterranean, and has really been a windy road. But I always find that you take one step forward, and whenever you are presented with a really cool opportunity, you never say no, and you just keep going. Um, I don't know. It's there's been not much of a plan, but the plan has always been go where your interests lie and where the funding lies as well, which is pragmatic approach to it.
Yeah, and so for me, I also started my journey in Canada, and then um, moved across the Atlantic to Europe, where we both are now. Um, and I guess I didn't do as much jumping around, or doing as much field work as Anish has done, but I think just I was always interested in research that was related to urban environments. So I kind of sought out urban areas and people doing work on urban aquatic ecosystems, which you'll understand more when we start talking a little bit about the project that we're going to present today. Um, Otherwise, I don't know. I don't really know what to say about jumps, but moving
The biggest
from
jump
the
was
biggest jump was moving from after my PhD, moving across the world to Sweden, where we are now. So that was a really big choice to, leave my family and everything that was in Canada and move to Sweden. Luckily, Sweden's not too different from Canada in terms of weather, but yeah, it's been a really exciting but also challenging time moving to a country where you don't know or speak the language and setting up a whole new life here. But Sweden's been very kind to me, so it's been great.
yeah, exactly. I guess we should emphasize our biggest jump was from across the Atlantic between our PhDs and our postdoc years. Um, uh, we left Canada to start postdocing around Europe. That was both exciting. It was stressful, but it's led us to where we are now.
Yeah, I use the word, thank you for that answer. I use the word jump maybe so people don't think that they have to provide us with, an exhaustive bio. but but I
Yeah.
can see words matter. And so, hooray, Svelja, jättebra. That's a
Jättebra!
big,
Oh,
yeah,
vi pratar
it's a
svenska.
hmm a bit. That's a beautiful place to be, and these are really wonderful and exciting answers to hear about a little bit of what you've both done and what motivates you and the importance of following the motivation, and as you say, also the funding, and that everything that we do shines back onto what we're going to do in the future, and I see it shows that all of those jumps did inform what you're doing now and then bring you together to do this work. We love to hear about collaboration here. We try to be a kind of collaboration with bringing science to the people and breaking down barriers of access to science and access to meeting people like both of you. So thank you. And at this point, I will hand you both the mic, and your PDF has been pinned by Katarina, so everybody can get into that and follow you along. And then we're here to help field questions. People might put questions in the room chat that we can share with you, and also to bring up friends later who might want to do a Q&A following your talk. So thank you so much.
Great. So
All right.
if people have questions, will you just interrupt us and let us know,
Yes, we can do
or
that.
will
No,
we?
it's up to you. You get to choose. It's guest's
Oh,
choice,
either
and you get
either
to choose if the questions drive your discussion. That's great. If you want to wait, it's all great.
I would say
Mm.
either is fine. We probably won't be watching the chat as much. So yeah, it's whatever works. People, you can interrupt if it makes sense at the time, and if not, we'll wait and answer them at the
Okay,
end.
that's what we usually do. And
Yeah.
you don't have the responsibility of monitoring the room chat. That's Katarina and I are here to do that.
Okay,
All right.
great.
So thank you.
Um,
The mic is
Great.
yours.
So we have a few slides prepared, and so when we go through the talk, I guess we will just shout out what slide number is relevant, just to keep people um, on the same uh, pace with us. And the first part of the talk, Aaron will give, and then I'll take over for the second half.
All right. So as I've been introduced here so far, my name's Erin McCollum, and I'm a assistant professor. And what I do a lot of research on is ecotoxicology, meaning the study of contaminants in the environment and how they impact the environment and the animals that live there. So starting off this talk, we look at the first, I guess it goes from slides 2 to 4. I have a few, it kind of builds on itself, but this might be what a typical household cupboard would look like in a home in North America or Europe, perhaps, where you have a cupboard somewhere that's stuffed full of medications that you might use or different uh, cleaning products. And we use these kind of products quite frequently, sometimes daily in our lives. They can include things like pain relievers that you use to treat a headache, some allergy medications that you might use during the springtime, or different types of prescription medications, as well as things like antibacterial soaps and detergents. And altogether, these compounds are referred to as pharmaceuticals in personal care products. And the use of these products in developed countries is really common and widespread. If we look at slide 5, this graph is showing the production and use of chemicals over the last 50 or so years, scaled to the year 1970. So it really shows that our use of chemicals is on the rise. And I've put some purple boxes here around the number of both approved pharmaceuticals that have gone on the market in the US and global pharmaceutical consumption, which has been growing. And one of the biggest point sources of how these chemical compounds get to aquatic habitats is through us consuming them and then traveling to wastewater treatment plants. So if we look at slide 6, this is the primary pathway that we'd see, where we use a pharmaceutical, we take a pill or something like that, but the dose that we're prescribed or the dose that we consume isn't fully absorbed by our bodies. So some of it is excreted back to a toilet where it's flushed down the drain and makes its way to a wastewater treatment facility. And this facility will treat that water before it's discharged back to surface waters. And when I say surface waters, I mean the water we can see on the surface of the planet, so rivers, lakes, the ocean, and so on. What happens in a wastewater treatment plant and the water use cycle is actually quite complex. And so on slide 7, what I'm showing is the wastewater treatment cycle. Wastewater treatment plants collect water waste from homes, businesses, road runoff, and sometimes even industries. And it makes its way to a wastewater facility where it's going to be run through several steps of treatment to remove and break down contaminants, bacteria, and nutrients, allowing the water to clarify. So after this, wastewater treatment plants might use several final cleaning techniques. So I've shown that here with the little electricity bolt showing UV sterilization, or using sand or charcoal sterilization. This is all to just make the wastewater as clean as possible before it's put back to surface waters. And when we discharge wastewater back to surface waters, we call it wastewater effluent. So pharmaceuticals really go from being on the shelf in our homes, through our bodies, through a wastewater treatment plant, and back to surface waters through this process here. And while the pathway that I've just described through wastewater treatment effluents is the main pathway, there are many other ways that pharmaceuticals can enter the environment. And some of these pathways are described in and shown in the infographic on slide 8. So this comes from the OECD, or the Organization for Economic Cooperation and Development, which is an international organization that conducts studies like these. And so they're showing some of these other pathways, which can include through aquaculture or the veterinary treatment of livestock. You can get runoff from the improper use and disposal of pharmaceuticals in landfills, or even runoff from fields that are receiving manure that contains pharmaceutical products. So there's lots of different ways that pharmaceuticals can get back into the environment, but the main way is through wastewater treatment plants. And so wastewater treatment plants are a nice kind of schematic of them is shown on slide 9. They tend to look like this, very industrial looking. But the main thing I want to point out, and maybe that you've already kind of deduced, is that wastewater treatment plants aren't able to remove all the chemicals that flow to them. So on slide 10, I'm showing that there's lots of different chemicals that potentially enter a wastewater treatment plant. But these chemicals aren't really able to be removed, and that's because wastewater treatment plants are kind of an older technology, and they weren't really designed to deal with modern synthetic chemicals. Wastewater treatment plants are really great at breaking down things like nutrients and bacteria to make the water safe. But they're not as good at fully removing contaminants. And so here I show different types of chemicals that can enter from a wastewater treatment plant effluent. But today on slide 11, we're really going to focus on pharmaceuticals. And that's the focus of a lot of my research over the past decade. Pharmaceuticals have been measured in the environment on a global scale. So on slide 12, I'm showing the results from a recently published study, it came out in 2022, where they measured pharmaceuticals at different sites all over the world. So all these little spots on this map are places where they took samples. And this research team found pharmaceuticals on every continent on Earth, even Antarctica. So pharmaceuticals are really a global problem when we think about them as novel environmental pollutants. And when they get out into the environment, what happens? On slide 13, what I'm showing you is that when fish are exposed to pharmaceuticals in wastewater effluents, they are able to absorb pharmaceuticals from that wastewater effluent into their tissues. So these are results from a study showing that pharmaceuticals absorb into different types of tissues, like the brain or the gonads, when fish were exposed to different dilutions of wastewater effluent. So pharmaceuticals are getting out into the environment and they're getting into animal tissues, but what impacts do they have? And for our summary so far on slide 14, what I can say is that hundreds of different pharmaceuticals have been detected and measured in the environment to date, usually at lower concentrations. So I'm talking nanograms to milligram per liter ranges. They can be measured in many different parts of the environment, but today I'm focusing on the aquatic environment. It's a global problem, and it can impact different biological systems. So this is where I'm getting at what's the ultimate or the downstream effect of animals being exposed to pharmaceuticals in the wild. On slide 15, our main concern is that pharmaceuticals are designed to affect human physiology. So pharmaceuticals act kind of like a lock and key. When you take a pharmaceutical, it has a specific drug target, like a receptor or an enzyme that it's going to interact with and have some sort of downstream effect in humans. So you can kind of think of it like a lock and key as an analogy, where the pharmaceutical is the key and you have some sort of target in your body that's the lock, and they fit together and create some sort of action. But these drug targets that I show on slide 16 are actually quite well conserved across the animal kingdom. So if we focus on the right side of this infographic here, of the 549 drug targets that these researchers looked at in humans, they found that 503 of them were present in fish as well. Slightly fewer, so 352 were present in Daphnia, which is a type of aquatic invertebrate, and even lower in another aquatic invertebrate species. So they're not perfectly conserved, but they're still well conserved, which means that a pharmaceutical that has a biological effect in humans could possibly have the same effect in non-target organisms like fish and aquatic invertebrates. And on slide 17, it's harder to see on this slide. The pharmaceuticals are designed to modulate human physiology and sometimes even human behavior. If we think about psychiatric pharmaceuticals like antidepressants or anti-anxiety medications, might they also modify the behavior of fish and aquatic invertebrates that are exposed to effluents in the wild? So this has been kind of a more recently explored question in the field of research that both Aneesh and I are working in. And it's an important one to ask, and I hope that we've started to answer it with our study that we'll talk about today. And I'm going to hand the mic over to Aneesh now, because he's going to talk a little bit about why we should study and care about behavior as a response.
Okay, so we'll move over to slide 18 now. And I just wanted to reiterate that Aaron and I have been working as a team for quite a while now, and this team is really comprised of Aaron, who's an ecotoxicologist, and myself as an animal behavior researcher or a behavioral ecologist. And so in a lot of the studies that we've done together, where we look at the effects of pharmaceuticals on wildlife, we choose to focus on behavior as an endpoint. And this is for a number of different reasons. Firstly, because pharmaceuticals, as Aaron just mentioned, are designed to affect physiology and behavior in humans. And so it's likely that they can also have effects in animals as well. But we also study behavior because it's an extremely important uh, trait for survival and reproductive prospects in animals. And so this is essentially their evolutionary fitness that we're talking about. I mean, if you don't behave very competently in the wild, then you won't be very successful at surviving or reproducing. And behavior is also very important if we want to understand processes that affect higher levels of biological organization. And so what do I mean by this? Well, if you look on slide 18, um, we have a diagram here that shows how life on Earth can really be represented as a structured hierarchy of physical, chemical, biological processes that are all building upon one another. And so you can see this on the slide, the hierarchy of biological levels that start out with the very small microscopic levels, going up to the whole organism level, and then up to populations, communities, and ecosystems. And so at the very low levels, we can study things like how molecular interactions affect the functioning of cells and tissues. We can go a bit higher and study how tissues and organs interact with one another via maybe the endocrine or the nervous system to influence the health and well-being of the whole organism. And then at even higher levels, now we can start to study things like, oh, how multiple organisms live and interact together to form populations, or how individuals and populations interact to form communities, and so forth. But importantly, it's through behavior that organisms interact with one another in their environments. So if we want to understand, say, population dynamics, or if we want to maybe sustainably manage our environment and the ecosystems within it, well, this requires a deep understanding of the behavior of the animals living in these systems. So what happens when a habitat becomes polluted with pharmaceuticals? For example, if they're dumped into the environment by a wastewater treatment plant. Will this change the way that the animals behave in these environments? And does this have any consequences for the populations and communities of organisms that are living there? So let's go to the next slide and imagine, for example, that pharmaceuticals can change the natural behavior of animals. It can either change the behavior themselves, or maybe it'll make it so that the animals can no longer express the behaviors in the correct contexts in the wild. Well, if this is the case, then exposed animals may no longer be able to find mates or social partners. They may no longer be able to care for their offspring as well as they should. Perhaps they're not able to live together in groups as cohesively as otherwise, or maybe they simply, if they're exposed to pharmaceuticals, they no longer pay attention to their surroundings or to the dangers in their environments. And what this all means is that individuals exposed to pharmaceutical pollution, well, maybe they're not able to respond to the challenges of their daily lives anymore. For some, that might mean the difference between life and death. And for others, well, maybe it's the difference between being able to reproduce versus not being able to reproduce. And yet for others, perhaps it's the difference between simply raising one baby or raising 5 babies. So the degree of severity might differ from one case to another, but in theory, changing the behavior of individual organisms can have a number of impacts at various scales of biological organization. So we are particularly, or were particularly interested for this study in one type of behavior, which was predator-prey interactions. And you can see a couple picture examples of this on slide 20. So these are the interactions that occur when one individual attempts to capture and eat another one. Essentially, one individual is running for a meal and the other one is running for their life. And these are incredibly common interactions in nature. You know, nature is a pretty grim place. Animals eat each other all the time. And predator-prey interactions are extremely important for a stable ecosystem. So we started wondering, now we're on slide 21. So we started wondering whether pharmaceutical pollutants could affect the ways that predators hunted their prey, or maybe the way that prey tried to escape from their predators. So imagine what might happen if an environment is exposed to pharmaceuticals, and this pollution caused predators to maybe be more efficient hunters, or maybe less efficient hunters, or maybe it makes prey better able to escape or worse at escaping um, their hunters, their predators. Any of these changes could conceivably affect species abundances. It could restructure entire communities. So we wanted to set out to run an experiment to really look into whether pharmaceutical pollution could affect predator-prey interactions. On slide 22, we'll see what the study system was that we decided to work with. We needed a species of predator and a species of prey for our experiments, and we chose the damselfly and dragonfly system. So this, at first, this might seem like a relatively odd choice, because for most people, when you think about dragonflies or damselflies, you're not really thinking about top predators, right? Um, and when most people think about dragonflies and damselflies, they're probably thinking about the adult forms that you see on this slide. But what we chose to work with are their larval forms, which look very different, as you'll see on slide 23. So here we see some pictures of a dragonfly nymph, also a dragonfly larva on the left, and a damselfly larva on the right. So while the adult forms fly through the air, the larval forms are aquatic, and they live in freshwater lakes, streams, ponds, etc. And in this case, really the lives of the adults, they give off a vibe of being very peaceful, and it really belies the nature of the lives of the larvae. Life as a larval dragonfly, or especially damselfly, is hell. I would say it's incredibly dangerous, and a lot of things want to eat you. And in this case, in the case of these two species, it's the dragonfly larva that's the voracious predator of the damselfly larva. So dragons really love to eat damsels. But the damselflies have a very cool defense mechanism up their sleeve that they can use against predators like the dragonflies. So on slide 24, I have to apologize a bit for the low resolution of the diagram. It was taken from Encyclopedia Britannica, but it has a good illustration of how dragonfly larvae hunt. So they hunt their prey by projecting their lower jaws out really quickly towards their prey to try and grab hold of them. So they're really ambush predators or sit and wait predators, and they lunge out very quickly to try and grasp their prey. On slide 25, we see that damselflies have this interesting defense mechanism. And this is that they have three appendages at the end of their abdomens, and they look like leaves or paddles at first, and these are called caudal lamellae. So these little appendages, they help the larvae swim through the water, and they can also be used as gills to help them breathe. So overall, they're extremely useful, useful little traits. So when a dragonfly larva grabs hold of a damselfly by its lamellae, the lamellae can actually break off. So this allows the damselfly a chance to escape, which leaves the predator behind simply holding on to the lamella. So this process of breaking off is in what we call in biological terms autotomy. So this means the self-amputation of an appendage, and it's usually in the context of trying to save oneself from a predator. So autotomy is something that's evolved actually a whole bunch of different times in many different organisms. And a rather well-known example is in some lizards, like some geckos and skinks, they're able to shed their tails when they're attacked by a predator. So this is you can imagine this is a pretty extreme thing to do, right? To sacrifice a piece of your body, but in return you get to live a little while longer. For our experiment, we found this to be extremely useful. And this is because autotomy leaves behind essentially a countable record of failed predation attempts. So this means that we can put a predator and a prey together, and we can check on them periodically. Each time a damselfly goes missing, this means that the dragonfly successfully captured their prey. But each time a lamella goes missing on the abdomen of a damselfly, this means that the dragonfly was unsuccessful in capturing their prey. So this gives us a way to quantify both successful and successful predation attempts. So on slide 26, we can see the graphical abstract of the paper. And what we did was we captured some wild damselfly and dragonfly larvae local to where we are in northern Sweden. We brought them to the lab for some behavioral trials, and we exposed them to two different pharmaceuticals, cetirizine and citalopram, as well as their mixture. So cetirizine is an allergy medication. It's an antihistamine. And citalopram is an antidepressant. It's an SSRI. It's a selective serotonin reuptake inhibitor. And these are both very common drugs taken by many people around the world. They're commonly prescribed, and therefore they're also commonly found in surface waters, especially near wastewater treatment plants. So we took the insects from a pristine, a clean environment in the wild, and we exposed them to these pharmaceuticals in the lab. And we exposed them at concentrations that would reflect what we could expect to measure in some wastewater polluted environments. So on slides 27, 28, and 29, you can see a few pictures showing the procedure that we went through. And we were, I would say, very fortunate to have such a beautiful location to work at, to collect our animals from. These pictures are of a lake near to Umeå, which is the city in Sweden where we are based. And on the right, you can see one of our team members in chest waders collecting bugs using a dip net. And so the dip nets bring back lots of different creatures that we then have to sort through on the shore. And we're sorting through everything for the two species that we're interested in, which in our case was the northern damselfly and the common hawker dragonfly. So we transport animals back to our lab at the university, and we'll keep them in special climate-controlled rooms. So these are rooms where the temperature can be held at very specific levels, and we can also control the light cycle, so we can simulate sunrise and sunset. And this essentially lets us keep the animals in a temperature and a light environment that matches where they came from in the wild. And what you see here is also a variety of different aquariums, lighting stands for camera recording, and different sized tanks for running our trials in. So on to slide 30, our project involved placing the animals into two scenarios. So in scenario one, we put a single predator, a single dragonfly, in with a single prey into an arena together and tracked the movements of the dragonfly predator. So we wanted to know if the predator would be successful in capturing their prey or if the prey was successful in evading the predator. And we actually used an object detection tracking program to do this. Slide 31 shows the first of our results, and I have to say right away we found some very striking effects of the pharmaceutical treatments. So when they were exposed to the drugs, the predators all moved significantly more within their arenas. So on the x-axis here, you see the different exposure treatments that the bugs were exposed to. And on the y-axis, you see a measure of how much they moved around in their arenas during the trials. So whereas control bugs were more sedentary, the exposed bugs were really active. And so activity rate is a very general measure to take, and it's one that's pretty commonly measured in a lot of behavioral ecotoxicology studies. And what it tells us is that something interesting is going on. So there's a lot of commotion going on in these arenas. And to find out what this is, we have to dig a little deeper. And so on the next slide, we see another result that shows how cetirizine seems to make it less likely that a predator would actually capture its prey. And so here you're seeing survival curves for the prey organisms over time. At the beginning of the trials, we have, of course, 100% of the prey alive, but at the end of the trials, some groups had only 50% of the prey alive, while others had around 70. And so in the cetirizine exposure group, the prey had a much higher chance um, of survival than in the control group. And also in the mixture group too, you'll notice that the mixture exposure also had relatively or elevated survival rates compared to the controls. So in our first experiment, we're seeing an effect where pharmaceuticals are increasing the general activity um, of the predators and the prey. And for cetirizine, we're seeing that predators seem to be worse at capturing their prey. But as we're about to see, the story does get a little more complicated. So on to slide 33, we see the setup for the second scenario in our project. So here we placed a single predator into an arena, but then placed 8 prey items with them. And in these trials, we checked the insects periodically over a 24-hour period. So here we could count how many prey had been eaten and, importantly, also how many lamellae had been autotomized. So remember, this is a record of successful and unsuccessful predation attempts. So slide 34, we're seeing some results from this scenario, and this time our results look very different. So it looks here that cetirizine actually made it more likely that the damselflies would be eaten relative to our controls. On the x-axis here, you see treatment group again. On the y-axis, you'll see the proportion out of 8 prey that were eaten over 24 hours. So in this case, both the cetirizine and mixture groups had higher mortality rates compared to controls. So it's really the opposite of what we found in the first experiment. And a very interesting result is now on slide 35, where we found that lamellar autotomy occurred more often in the citalopram group. So what this means is that the damselflies in this group were able to evade the predators more often using their ability to self-amputate this appendage. Slide 36 now, well, what does all this mean? Well, it means that pharmaceuticals, at least the ones that we investigated here, can affect the way that predators and prey interact with one another, but perhaps not in a very clear and easy to understand way. And this is perhaps a very common occurrence in science. Things always tend to be more complicated than you first expect. The drugs we tested, particularly cetirizine, had this effect where they either made it harder to catch a prey or easier to catch prey, depending on the experiment and the social context. And we did find that in one experimental treatment, the predators seemingly became sloppier or less efficient, as indicated by the increased rates of lamellar autotomy. And so what this suggests is that polluted habitats can experience altered predator-prey relationships, and this makes it harder to predict how species communities will be structured in impacted areas. So we think that this is a very exciting area of research, and we're hoping to expand on it in the near future. We will leave it at that, I think, for the slides, and I think we're happy to take some questions now.
Yeah, and you'll be able to see there's a few more slides here, but there are kind of some extra ones we have that we might use for certain questions that might get asked.
Well, thank you so much for this wonderful presentation, and I think for this really important work that you're doing, and that you're taking the time to share this with us in such a really interesting way. So first of all, thank you for that. And yeah, everyone that has a question, please go ahead and raise your hand or post comments in the chat. If you yeah, if you cannot, speak up for now. And And in the meantime, I saw that Tom had the question
Hmm.
in there, but I feel like you kind of answered that. I don't know. Do you see the chat in terms of cause and effects?
Yeah,
Uh,
it's an interesting
yeah.
question. It's about whether the the actual pharmaceutical is what's driving the behavioral effects, or maybe a breakdown product of the pharmaceutical within the organism.
Yeah, and I can answer that. Well, I can try to answer that. A lot of pharmaceuticals actually have bioactive metabolites, like Tom has pointed out here. So sometimes pharmaceuticals break down, and it's the thing that they break down into that is actually more biologically active than the parent compound or the original compound. I can't say without doing a bit more research for certain about cetirizine and citalopram, what their ratios are. I know for a few others that we work with commonly, we don't, at least at this moment, know of any uh, bioactive metabolites, and we didn't measure any in our study. But in some of our other work, we've looked at the metabolites of, for example, fluoxetine, which is another SSRI antidepressant, or some of the benzodiazepines that break down into other active compounds. So it was not the purpose of this study to answer that question. This was mainly focusing on the parent compounds and whether they cause behavioral change. But the question about their breakdown products is really at the forefront of the field, and other researchers in our lab are actually working on that question.
Hmm.
And if you're interested, I could definitely share some links about that.
Yeah, Tom, I see you here. Did that answer your question, or did you want to follow up?
Yes, it did. And thank you very much. It's been a really interesting and wonderful presentation, and really makes me wonder also is that if we have, say, a pharmaceutical effect on the behavior of a prey, how might that affect the predator and the predator's predator, and etc., etc. So that really goes up the food chain. And it amazes me, but also scares me to wonder how different pharmaceuticals could effect different changes, especially on potentially important animal groups like insect groups like the bees that we rely on for pollination, and also the food that we eat. So, how might you envision that some of these potentially recalcitrant pharmaceuticals might actually significantly affect, say, food crop production, or even how the microbes in the environment might be involved superficially in global change, etc. Thank you very much. Appreciate it.
Thanks for the question.
It's a very deep
It's
question.
a deep, and there's questions within questions there. Um, what I can say a little bit is that you touched on microbes, for example. So wastewater effluents discharging antibiotics into freshwater environments is a big concern for the development and evolution of antimicrobial and antibiotic resistance. It is not an area that I currently work in, but there are other world-class researchers that are focusing on this question specifically. Yeah, and if you were really interested, you could email me, and I could get you some contact information or websites of people who work in that area that could answer your questions. Um, as for the bees, that's an interesting question, and I don't know of any studies off the top of my head that have looked at how pharmaceuticals, for example, in water, might affect bee behavior. A more pressing, or not pressing, but a more commonly studied question is how different pesticides affect bee behavior. There's a lot of work done showing that different neonicotinoid pesticides are bad for bees, that they impact their learning or their ability to remember where they forage for food during pollination. It would be really interesting to study how different pharmaceuticals might affect bee behavior. We'd have to design some lab studies to do that, or do some field sampling. But one thing I can just say is that the idea of that you kind of touched on here, the transfer of pharmaceuticals from aquatic environments to terrestrial environments, is really at the forefront of this research field right now. Me and some researchers have recently done a study here in Sweden where we looked at the transfer of pharmaceuticals from water to riparian spiders. So, spiders that live and forage along aquatic water bodies. And we did find certain pharmaceuticals tend to bioconcentrate in spiders more than others. There were different antibiotics as well as anti or psychiatric medications. As how it would affect crop production or food production, I can't say with much certainty right now. But another thing to think about is that when wastewater treatment plants create biosolids, so the solid waste that comes from wastewater treatment plant, some of that gets spread back onto farmland. So I know that there is research now being done, for example, on if you put biosolids on a field that's growing tomatoes or lettuce, how much pharmaceuticals that are in those biosolids end up in lettuce and different vegetable products. So that's something that's being actively researched right now, but I don't have any concrete kind of take-home snippets of results. It's all very complex and still being researched.
Yeah, and an important thing to note is that everything in the wild is really being exposed to mixtures, and so we do our experiments on one or two compounds, and we try and figure out what effects that might have, um, and whether that translates to effects in the wild is a bit of a different story, which is why we are striving towards doing more work on mixtures or using wastewater effluent itself, which is a mixture, to expose our test animals to figure out what effects it'll have. But it gets quite almost overwhelming once you start thinking a bit much about the complexity of the uh, mixtures that animals are getting exposed to in the wild, and then also the rippling effects, like you said, that are going on across trophic levels. Um, Not only rippling effects, but these reciprocal effects through social interactions, where one individual whose behavior is perturbed will affect the behavior of another individual, and vice versa. And this can change selection pressures that animals face and can change the evolutionary trajectories of whole populations. But um, yeah, the questions are seemingly endless. Maybe we can.
Yeah, I saw there was another question here from Deepak on what are the initiatives taken by pharmaceuticals to minimize the effects on aquatic ecosystems? And that's a really interesting question because we by no means are advocating that people should stop using their pharmaceutical medications, especially if you need them for various illnesses. Definitely don't stop using them. They are miracles of modern medicine. But there's kind of two approaches to removing pharmaceuticals from or preventing pharmaceuticals from entering the environment. And one could be called an end of pipe solution. So that's something like upgrading wastewater treatment plant technologies. So there's more modern types of wastewater treatment that are better at removing pharmaceuticals before they're put back into the environment. However, those cost a lot of money. And so then they're a big infrastructure investment that municipalities have to make, and that ends up costing taxpayers a lot of money. So it's very contentious, a very contentious topic in a socio-political perspective. Then there's kind of the before pipe solutions, which might mean engineering or designing pharmaceuticals to break down more quickly so that they have less of a lasting effect. That's one idea. or also changing prescription patterns. And now I said that you shouldn't stop using the pharmaceuticals that you need to use, of course. But a good example of this is how there's been a recent shift in not over prescribing certain pharmaceuticals like antibiotics so that we don't contribute to the rise of antimicrobial resistance. So those are the kind of two perspectives on how we can deal with pharmaceutical pollution. Do we either stop it at the end of the pipe just before it enters the environment, or we make changes in our consumption and design of pharmaceuticals so that they have less of an effect and don't need to be removed before they hit the environment?
Yeah, and I can say that a little plug for the institution where we're at, the Swedish University for Agricultural Sciences, SLU. There are researchers here at the university who are working with wastewater treatment plant technologies and trying to design new technologies specifically for removing different classes of pharmaceuticals and personal care products. So it's on people's minds. We're getting engineers to try and tackle this this problem. Is there another one?
I have a question, if you got some time.
Yeah.
So, back when I was in high school, I used to try and get out the city. I'm from Chicago during the summertime, and one the things I used to do was go to these little research programs at University of Illinois, and they have a pretty big agricultural school up there. And this particular summer, the project that I was kind of tasked with or got to help out with um, had to do with the differences in the actual fauna in these different microenvironments in different streams that may be up or downstream from agricultural places where there would be runoff. One of the things we saw was there was differences in the actual invertebrates that was there. So I was kind of curious to if you guys did or even could kind of see if there is a difference, excuse me, if there's a difference between the actual fauna between places that are, you know, uh, pharmaceutical runoff or waste versus agricultural runoff and waste and so on and so forth.
Yeah. Uh, yeah, I mean you're describing one of our research projects right now. Uh, this is a big program that we have going on, or it's spearheaded by Erin. It's really her one of her academic babies. So I'll let her describe it.
Yeah, so right now I'm sampling um, fish, fish communities and aquatic invertebrate communities at wastewater treatment plant outfalls upstream and downstream to look at how these inputs of like complex chemical stressors affect biodiversity in these environments. I don't have any answers for you about this study right now because I'm still collecting the data, but in theory, wastewater treatment plants tend to discharge nutrients like nitrogen and phosphorus that can enrich the environment around an outfall. So that might mean that they're able to attract organisms to these sites. So you might have more aquatic invertebrates or more fish actually at an outfall where they might be potentially exposed to harmful pollution and they can't tell that. But it all depends again on how toxic or how dirty the effluent is, if you will, because there'll be some threshold where it won't be conducive to supporting aquatic life. The nutrients will be too high, it'll be devoid of oxygen, so then animals won't live there. So it really will depend a bit on the treatment level of the wastewater treatment plant, and then yeah, the animals that tend to live in those environments to begin with. So it's definitely a great question, and it's something that people are thinking about. And the program that you did sounds really interesting. It
Mm-hmm.
would be cool to learn a bit more about the researcher or the research group you were working with there.
Yeah, the what we're describing here, the attractive force or the potential for some of these point sources of nutrients and pollution to actually attract animals and change the community around them is what's called an ecological trap, or at least that's the how we're thinking about things. So it's a kind of abiotic conditions that really are attractive to animals, but then when they've been attracted to a certain area, they're then exposed to some deleterious factors as well. And so we are envisioning, where we're hypothesizing that things like wastewater treatment plants can act as these traps in the wild. And but what we're looking for in this research program is change in differences in species community, fish communities, insect communities, downstream and at the outfalls of these wastewater plants. And unfortunately, we don't have any straight answers for you yet because the data has not been analyzed yet. But it is currently being gathered, and it's a project that it's an ongoing long-term project. So we're hoping to really dig into the data in the coming year or so.
I have a follow up question, and also before I get to the follow up question, I'll see if I can reach out to that project head because essentially it's a the whole purpose of that program is to get like at risk youth from inner cities pretty much across the globe. So, I came from Chicago. We had people from Puerto Rico and some people even from Greece that would come over to take part in the program. I'll see if I can contact them to see if they can find out which lab is responsible. This is shit,
Mm-hmm.
man, 15, 16 years ago. So it may not even be the same person in that department, but I can see if I can contact them. And I followed you.
But also,
So if I,
no worries, no worries if
okay,
you can't do it. It sounds like
but
a super interesting program, and I'm really glad you were able to partake in something like that.
Yeah.
yeah, I mean, even so, like collaboration in the scientific field to me is always a beauty. But my follow-up question, um, is kind of sounds like that sometimes when you get these newly polluted environments, that it can cause a change in the microenvironment of whatever space, like in this regards, different water systems that could almost cause like an ecological hole. And like now my question is like, do you think it's worth investigating if there is ecological hole, if that's what makes like certain invasive species so easy to invade? Because the reason why I
Mm.
asked that, being from Chicago, we have like these little water systems that go through the downtown area, and the water is dirty as all hell. And one of the issues we have is invasive species. And it kind of makes me wonder if the lack of diversity uh, ended up making the hole for these invasive species to then come in and thrive.
Yeah, I think you hit the nail on the head there. The effects that these environmental stressors can have, so these chemical stressors, can be to reduce the biodiversity of a native ecosystem. And one of the possible consequences of a reduced biodiversity in an ecosystem is that its biotic resistance goes down, and that's essentially uh, what we call how resistant an ecosystem is to being invaded by a non-native. So it's certainly possible that by damaging or reducing the diversity of a native ecosystem, you make it more susceptible for invaders to come in and take root and to proliferate. So it's certainly very, very, very plausible.
Yeah, and,
Um,
and we've even, in my PhD research, we even saw that happen. So I did my PhD research in Hamilton, Canada, which is on the edge of Lake Ontario, um, and a very, like, kind of like Chicago, super polluted city. It has historical steel industry, three different wastewater treatment plants discharging into one harbor, and so on. But we found that when fish communities near our wastewater treatment plants were really driven by one invasive species that was super common. So it seemed like kind of an empty habitat that this invasive species decided, yeah, this looks great. And because they tended to have traits themselves that allowed them to live in a less desirable area, they were able to exploit the empty habitat. So
Yeah, like
yeah,
they tend to be extremely tolerant to a wide range of conditions, and
yeah.
uh, many native species are less tolerant, and so these invaders tend to outcompete them in these already stressful environments.
Chicago is a super interesting area too because you've got kind of the connection point between the Great Lakes water system and the Mississippi watershed, and there's a lot of efforts being put in Chicago to prevent the spread of invasive species both from the Great Lakes to the Mississippi and vice versa. So it's really an interesting area to do research like that.
Absolutely. Thank you for answering my questions.
No problem. Thank you.
Yeah, thank you
Do
so much for all these questions. And I wanted to check with you how much time you still have because we've been going over an hour. So I wanted to,
Oh, we're
be
happy.
respectful
It
of
doesn't
your.
matter. We're happy to
we could
answer some
stay a
more
bit longer.
questions if there's still more. Whatever you guys as moderators prefer.
Um, so is it, Deepak asked in the chat, is it possible that excise dumping of pharmaceutical waste to aquatic ecosystem leads to mutation or extinction of certain species in the next years to come, or is it already happening?
Extinction, yeah, that's a very extreme outcome, and we can point to certain extreme examples where something like this has happened, like the vulture example. But
Yeah,
it's
but
not.
it will never be an extinction like a whole species extinction where they'd be removed from the face of the earth and you'd never see them again.
It'll be more. We're talking local
Local
extinction.
extinctions where their species used to be there, but then because they started adding contaminants, they're not able to live there anymore. So yeah, as Anish mentioned, the vulture example. So in uh, I guess this study was mainly done in India, but basically the cattle were being treated with an anti-inflammatory drug called diclofenac. And then when cattle ended up dying, the carcasses were left out, and vultures, which are a predatory kind of scavenger bird, they would come and feed on the carcasses. But vultures ended up being very sensitive to diclofenac, and they um, died after being exposed by foraging on these contaminated carcasses that were contaminated by being treated with pharmaceuticals. So that led to local extinctions of these. I can't remember which species of vulture, but a vulture in the area that this treatment was happening.
Mhm.
So it's something that can happen transiently in a specific kind of area, but we haven't seen at a continental or global scale by any means. And there's still the possibility that when you have a local extinction that you can have a reintroduction if you remove the stressor and either the species naturally moves back into its normal range or it's reintroduced by a restoration program or a reintroduction program.
Yeah, thank you. That is interesting. And I wanted to also add the question maybe the relationship to like the human environment. Do you feel like this is kind of a model, a bigger, more complex model system than researching this in the animals in the lab? And this could reflect maybe some mental health related problems or behavior, especially in evolving human, like a child, um, like that symptoms maybe occur more often or more prevalent because of this exposure in the environment, or don't you think that this would be the case?
Do you mean that that exposure to pollutants in the environment, the human exposure to pollutants from the environment is driving like changes in human behavior?
Yeah, exactly. So, in drinking water and so on, I would assume that there are also
That's
pharmaceuticals and so on?
that's definitely an interesting question, and I think it's hard for me to say. A better field of research to answer that question is environmental epidemiology. So that's more people who do research on the effects of chemical contaminants on human disease burden and occurrence on a like large scale. I know that there's been correlates, for example, that have been done when leaded gasoline and leaded paint used to be used, and IQ scores in children, for example. And then once lead was removed from gasoline and paint, that we saw IQ scores go up. But that's all correlation. It's hard to prove causation with relationships like that. And unfortunately, well, not maybe not unfortunately, fortunately, with humans, you can't put them in a lab and expose them to different compounds. You kind of have to rely on these correlational relationships. I don't know if that's a satisfactory answer, but it's definitely a possibility, but it's not something that I know of that's been studied so far.
Oh yeah, yeah. I would assume that is very complicated and really hard to do always everything in human, like what's the
Yes.
causation. But yeah, it's interesting to think of kind of the whole system there, and because we see so many, like such a rise in, you know, in mental health disorders, especially in youth and preteens and so on. And we make all kinds of links, but it's probably a huge mixture of stuff. But I feel like people care more um, to regulate these exposures if it's also affecting humans. I guess that's why I made the
Yes,
question. Like,
A
absolutely.
100%.
It's not until, yeah.
People care more about humans than the environment for sure.
yeah, so, like, I don't know, is there a huge variability of amounts of pharmaceuticals depending on where people live in which countries? Like, I would
Yes.
assume that in places where they prescribe just more would probably be higher exposure.
Variation.
Yeah, yeah. So there's a lot of global variation, and in that study that we showed with the map and all the little dots on it, that study also measured the concentrations of various pharmaceuticals in different countries around the world, and basically the highest concentrations are in countries that have developed enough infrastructure to deliver pharmaceuticals to the population masses, but they don't have well developed wastewater treatment. So this tends to be more um, middle income countries that are rapidly growing at the moment. You also, there's just a ton of variation in what pharmaceuticals end up in the environment based on the population that feeds the wastewater treatment plant. Is there a hospital connected to it? Is the population an aging population? There's also even variations on a weekly or a diurnal or a daily basis. You tend to see more pharmaceuticals, in the mornings and the evenings when people are at home. And you see certain variation, seasonal variation, for example, in pharmaceuticals being discharged, like what are they called? Antihistamines. Antihistamines that treat seasonal allergies. You tend to find those more in the spring and summer than you do in the winter. And even the seasons themselves can affect how pharmaceuticals in the wild will break down. So the amount of UV they're going to get will affect how they degrade in the environment, how warm it is. As with everything in ecology, it's complicated and it depends. So yeah, there's a ton of variation, like you say.
And on this note, there's also interest in using these technologies to quantify pharmaceuticals, but also illicit drugs in the wastewater of various towns and communities, basically to get an overview of the drug consumption um, going on in different areas, and so that's
Something
something
that happens.
that's happens.
Yeah, called wastewater monitoring.
Yeah, I would say that around Brooklyn, all fish are on probably shrooms and coke right now. I
Quite
don't see
possibly.
Yeah,
what the fish do there, but yeah, no, it's I think it's a very important research, and I read an article, it was a few years ago, that basically says exactly to those drugs, if you don't need a drug, just don't take it because it's bad for your ecosystem around you. So,
yeah, that's
yeah, I would
not
emphasize that.
something to live by or strive for.
Mm, definitely.
So, yeah, I think this is a very important research field. And is there also a lot of development and developing better and cheaper sensors for a better and larger scale, um, monitoring? Or is that something that people are not really yet thinking about, or governments, or, different districts, because it's people are not aware yet of that problem so much?
No, I monitoring programs are definitely something that people want, as well as different governments would like, and costs always are basically the bottom line. There is research. I'm sure that there's research going on about how can we monitor chemicals in a more cost effective manner, because right now we tend to use things like mass spectrometry to measure pharmaceuticals or the concentration of different pollutants in samples that we get, whether it be in water or tissues. But running a mass spectrometer costs quite a lot of money. They cost a ton of money just to buy to begin with, and they tend to be located in commercial labs or universities or government research institutions. So it's something that I'm sure is being developed, but at the moment there isn't any really easy solutions to it yet. And hopefully over the next, 20, 50 years, we'll see that gap getting closed.
Yeah, it's still a rather expensive endeavor.
Yeah, I imagine that it can be easy, and but yeah, I feel like the cheaper it gets, and also developing countries, and where the problem is maybe kind of very acute now, um would be able to monitor better and help with that, keeping this problem at bay, basically. um you would just be more affordable. So, um yeah, but I don't know enough of the field of developing those type of sensors. I know that there's a lot of development in personal sensors, on phones and stuff, to maybe, I read about, to maybe monitor blood sugar levels and alcohol levels and things like that. But I'm not sure if you could just use that, to develop also sensors that you could just stick in your water at home. I
Yeah,
don't know.
it's
That would be
yeah, it'd be super
tremendous.
interesting, and definitely something that would increase the access of lots of communities to be able to monitor pollution in their environment. So
Yeah, it would put a lot of different of these chemical pollutant problems more on the radar.
yeah, it's a problem that hopefully our engineers and chemists and so on will tackle in the future.
Yeah, I agree. Even if the absolute value is not, of course, not as accurate, but as long as it can tell differences pretty
Yeah.
well,
Mhm.
that would be really good. And are there filters or anything that actually kind of break down pharmaceuticals really well, or just, sunlight exposure is enough? Like, are any districts working on, somehow distracting or filtering pharmaceuticals out already?
Yeah. So some of the extra treatment stages that I showed in my graph, UV sterilization is one way to remove pharmaceuticals, as well as ozone sterilization. charcoal filtration and sand filtration are other ways to remove them. Then don't function maybe quite as well as ozone or UV. They're not just quite as effective. There's also something called membrane reactors, which I can't describe how those work because I don't know enough about them myself. But they're essentially a membrane technology that would absorb pharmaceuticals as water passes through them. Um, so these are all definitely have been researched and designed for use on wastewater treatment plants and are now being put out in at wastewater treatment plants. A great example of this is actually in Switzerland, where the whole population voted to have advanced treatment on wastewater treatment plants to remove emerging contaminants like pharmaceuticals. So at a national scale, they invested in the infrastructure to remove pharmaceuticals with these advanced treatment technologies. In Sweden, for example, on a case by case basis, um, these types of technologies are being implemented, but it's a slow process. Usually, an area has to first do a study to show that there is pharmaceutical pollution, and then they have to get the money together basically to be able to afford to do this kind of upgrade and implementation, which can just take time. And then once the money is recruited, it's another question of building the infrastructure at scale. So I think it's something that we'll see more and more, especially in developed countries moving forward that already have base level wastewater treatment plants functioning, and then they'll add on these advanced upgrades. Yeah.
Of course, Switzerland does it. I mean, you always feel like they do everything better than anyone else. Um,
Yeah, of course it is.
Yeah.
yeah, so yeah, I really hope that communities, governments will listen and then implement those. And I hope, that you continue getting a lot of funding and to do this research and maybe even, do more, because I think it's a really important part of pollution that gets ignored a lot. And um, is there a reason why you went to Sweden? Like, doesn't this get funded too much here on this part, or like in the
Yeah.
States or in Canada a lot? Like, is there a higher sensitivity in Europe for this?
No, 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 no. It all had to do with finding a postdoc. So at the time when I finished my PhD, I had applied to do postdocs in several different labs that were working on this area. Some in Canada, some in the U.S., and some in Europe. And it just, the way things broke down, that the proposal that we wrote, me and my postdoc supervisor Thomas Burden, was funded in Sweden, and then I moved to Sweden to do that project, and have been very fortunate to continue developing my career here since then. So it's a global problem, and there's researchers that do work on questions like this in the U.S. and Canada as well.
Mhm.
And yeah, the reason why I moved was the opportunity to work with somebody and have the project supported at the time.
Yeah, I'm glad
Yeah.
to hear that there's still, even after Trump, that there's still researchers here in
Yeah.
the US working
Yeah.
on this. So
There,
we still
there's there are these hubs for for researching different topics, and they don't necessarily have to be located in an area where that topic is a super high priority or problem, but it sort of is an expert in the field who's building a working group around them and invites lots of researchers there, and you just passively increase the working group, and you get these areas of expertise who happen to be in areas, in our case, in northern Sweden, which is far removed from a lot of population and a lot of wastewater, but still is very active in researching it.
Yeah, that's really interesting. And I mean, if you would want, or if somebody, you know, would want to transition to study this in humans, I think the public data sets are quite good in Sweden and Norway also to look into region specific maybe outcomes in health. I think a lot of public health research is based on a data set from these countries, so that would be another upside maybe for the future. Or is that correct?
Possibly, yeah.
Could be, yeah.
Yeah, there's also, I think there's lots of like long-term blood, for example, like blood monitoring data sets that are hosted in the US as well through the not NIH, is that or is that the UK? Yeah, maybe the NIH that have taken blood samples, for example, over decades and can measure the different pollutants in them in humans. But yeah, it's definitely a possibility. And perhaps there's more access here in Europe.
Well, yeah, what I think, as I said, this is really important, and I congratulate you on this research, and thank you so much for coming and sharing this with us. And I'll be really curious to continue following your work, because yeah, it's such a it's a field that is really complicated, and I always admire people that go into ecosystem behavior fields, because yeah, it's a high complex system, so it's really important. So thank you, and thanks for doing this
That's
research.
very rewarding too. But thank you for having us. It's been a pleasure.
Yeah, it's been so fun sharing our work with you today.
Yeah, and also everyone, thanks for posting comments, asking questions. And if people like research talks like this, like or similar, we'll have our next room about biomaterial and nanoparticles that were developed as a new method for spinal cord repair. So if you would be interested, come back. And yeah, thank you again, Aneesh and Aaron. Good luck for the future. And yeah, I hope maybe we'll hear you one day again. So thank you again.
Sounds
It was
great.
a pleasure.
Thank you.
Thank you for having
Thanks,
us.
everyone.
Okay, I'll close the room in 3, 2, 1. Bye, everyone. Thank you.